I'm super excited today to present this work because it was just such an amazing journey to conduct this work at the Icahn School of Medicine. And I hope that at the end of the talk, I can convince you and you are as excited as me about it. So I want to start with a question to all of you, kind of asking, do you think there's a bottleneck for the translation of microbiota therapeutics? Because I kind of want to share my vision, and I think there is a real bottleneck that has held back the translation of therapeutics in this field. And I think it's not the science, the problem is twofold. It's the overly costly GMP production with industry, and also the unclear pathway to the clinics that has so far limited the translation of microbiota therapeutics. And I now know that it doesn't have to be that way, and I kind of want to share you the vision I have for the future and what we have developed so far. So how do we want to open this bottleneck? We have developed a new platform for the manufacturing of defined microbiota therapeutics. And this platform is GMP compliant, but for a fraction of the cost you would pay in industry. We've also received IND clearance to pursue a phase 1 clinical trial. And the results of the trial are now available as preprint on medRxiv. But with the publication, does not only come the results of the clinical trial, it also includes the full IND documents and the master batch records for production, since we want to benefit a translation of microbiota therapeutics as much as possible by democratizing this knowledge. So today, my talk, in the first part, I'm going to tell you about the platform we developed and our first product. And in the second part, I'm going to tell you a little bit about the clinical trial. So I want to start with a little bit of an overview about what it take to go from stool to defined drug product. And here we start, as always, with a stool sample from a donor. And in the first step, you need to do an isolation and characterization of the single strain bacteria. And in the second step, you move to the actual manufacturing. And here we followed FDA guidelines on cell banking and manufacturing. So we first started with a research cell bank, which is pretty close to what we are used to in academia. And then we manufactured a master cell bank, a drug substance of the single strains. And those single strains can then be combined into the final drug product. This process and setup can be achieved with our current setup and platform in about 1 year. So what is the manufacturing platform? So we have a dedicated manufacturing facility, and it's all optimized for small space. So all you need is an anaerobic chamber. But it's not just a regular anaerobic chamber, there's a custom feature to that anaerobic chamber. And that custom feature is the sterilization port in the front of the anaerobic chamber. And this allows us to fog the interior as well as the equipment inside the anaerobic chamber with ionized hydrogen peroxide. And this let us complete nearly all of our production runs completely contamination-free. The actual fermentation of the bacterial strains is carried out in batch fermentations in single-use equipment. So the production is simple and robust. Despite the simplicity, we achieved very high cell densities for all of the 15 strains we produced in our first drug product, which allows us then in the final step, when we come to actually formulating our drug product, allows us to be flexible in the concentrations we choose for the strains and which combination of strains we mix in the final drug product. So the final drug product is obviously then mixed in pharmaceutical grade buffers. And we have so far produced over 40 drug products at very high cell densities. For the regulatory framework, which obviously is also very extensive, we have filed over 2,000 pages of batch records, and the release criteria for our drug product, you can see in the lower part of this slide, are a CFU count for potency, mass spectrometry for identity, and for purity, we followed Pharmacopedia guidelines, so USP methods 61 and 62, which are testing for specific bacteria, but also for total aerobic count of bacteria and also for yeast and mold. Before I want to transition to the clinical part, I kind of want to give you an idea, but I have to be brief here, on the design of MTC01, which is our first product. And this is a product to treat recurrent C. diff infection. And the design is based on engraftment data that we have from a previous FMT trial in recurrent C. diff infection. So what we have observed here is that the engraftment of the strains from FMT is very durable over a large time frame, a long time frame of up to 5 years. And what we have also observed is that the response does correlate with the engraftment success. So in principle, our design is based on the successful engraftment of strains in an FMT trial. So now we have our MTC01 drug product ready. So the defined drug product is in the freezer. We have received IND clearance from the FDA to pursue our phase 1 clinical trial. But we have also manufactured an FMT drug product. And I now going to explain when I move to the clinical trial part why we need that FMT drug product. So in the phase 1 clinical trial, we have for the first time compared a complex FMT drug product with a defined consortium that is a subset of strains only from that FMT drug product. So it's a real head-to-head comparison of kind of asking the question, is the subset of the strains that we observe to drive success of a clinical trial enough to recapitulate the success from the FMT trial? The clinical trial timeline looks like this. So you have an antibiotics treatment at first, and then you have a single administration via colonoscopy of either the FMT drug product or the MTC01, the defined drug product. And then you have a 26 weeks follow-up period. The primary outcome are safety and short and long-term relapse. And the secondary outcomes are engraftment, as well as persistence of engraftment, and also the effect on the recipient microbiome. So far, we have 18 patients dosed in the clinical trial, and we have seen no drug-related serious adverse events. We have very comparable success rates with the defined product and with the FMT product. And we have seen no long-term relapse. So we can say that we have confirmed and validated our platform so far. So we have safety and we have efficacy demonstrated by the drug product that we have produced with the platform. Coming to some of the scientific insights, which are actually the super interesting part. So we can see that the strain engraftment we see is best with our MTC01 high dose. So our defined product, and I now need to mention that we also have two different doses per treatment arm. So we have always a high and a low dose of the FMT drug product and the defined drug product. So we see that our high defined drug product achieves the highest engraftment. So we are only looking at the 15 strains that are included in the drug product. And we also see that strain engraftment is very persistent. So we see, as expected from, if you remember the slide I showed you for the FMT clinical trial, we saw that even over time frames of years, the engraftment is very persistent. But yeah, and here we so far have data for participants for 12 weeks, but it's going to extend to 26 weeks. And we see that now also the defined drug product has a very persistent engraftment. And we also know that we can confirm that we have picked the right strains. What we're looking at here is the frequency of engraftment. And every little gray dot here is one strain. And the frequency of one would mean it engrafted in every recipient. Yeah. And here we see only the high doses of MTCO1 and the high dose of the FMT drug product. And we can see again that MTCO1 has a very high frequency of engraftment for the 15 strains included. And the FMT high dose also has a very high engraftment for most of the strains and a somewhat lower engraftment for some of the strains. And here on the left-hand side, that's the interesting part. We see the FMT high dose, but strains that are not included into our drug design. So we see that we have a lot of bacteria strains that do not engraft in the FMT high dose, but we see a couple of strains that do actually engraft that are not included in MTCO1. And we knew that before already. And most of those strains are of the same species as a strain that is already included. And we didn't include them because we wanted to make the drug product as diverse as possible. And some of the strains also just have a negative safety profile, and that's the reason for exclusion. So as a summary, I hope I could convince you that we have developed a novel way to produce microbiota therapeutics, including the complete regulatory documents that we include in our publication. We have demonstrated safety and efficacy in our phase 1 clinical trial with MTCO1. And also we have shown that a design that is based on engraftment does work, at least in this case, where engraftment is probably a driving force for success in the clinical trial. And for the future, we are obviously still improving the platform, and we are working on a oral design for MTCO1. So we want to see if the success we've seen with the colonoscopic administration also does transfer to an oral administration. And we obviously are also exploring other indications for treatment. So if you are interested in this platform, then please feel free to reach out to me directly. And also on a personal note, as Eduardo just mentioned, I relocated recently to Switzerland. And I'm kind of working on figuring out how to best continue my work on this project. So if you're interested in building something similar or collaborating on this, then feel open to reach out to me directly. And with this, I want to thank you, and also a big thanks to the Icke School of Medicine and also everyone who was involved. It was really an exceptional project. Thank you.